Good morning. My name is Kelly Merc. I'm an adjunct associate professor at Duke University, and today I'm going to be describing the stable isotope resolved metabolomics project that we have evaluating normal stroma and desmoid tumors. This was a project that was started after a pilot study was funded by the Desmoid Tumor Research Foundation. The presentation today will cover the background information from the Untargeted Metabolomics project, and then I will describe the ongoing work that we are doing at Duke University to describe our study hypothesis, design, methods, early data, future data considerations, and data analysis and collection. So it is important to understand what metabolomics is, first of all. So 5 years ago, I would have had to explain the relationship between all of these molecules on the left, but it's pretty common knowledge now that we understand that metabolomics is an important technique for understanding systemic disease-associated phenotypes, in addition to any environmental considerations. Metabolites are mostly known and can be identified and quantified using several analytical techniques and through numerous sample types. I personally have evaluated blood products, cell extracts, tissue extracts, fecal water, urine, CSF, and whole zebrafish. So these experiments on the right describe the sample preparation and handling that's exceedingly important for determining differences in concentrations of these small molecules for metabolomics to yield useful data. It all starts with the experimental design and the question that we're wanting to answer. Then the sample preparation, generation on, again, multiple kinds of analytical instrumentation, and then the data analysis. The hope is that the phenotype in which we are interrogating will be strong enough that in an unsupervised analysis, we'll be able to differentiate the two populations through natural groupings to understand biological interpretations. In 2016, we received a new investigator grant from the DTRF to determine if metabolomics was an appropriate method for investigating these differences between desmoid tumors and normal fibroblasts. So we had access to samples from two patients, a T41A, S45F, with their matched normal fibroblasts. In addition, we did look at an unaffected fibroblast cell line from ATC. We found that not only the cell lysate from each one of these groups was highly reproducible by the analytical technique that we were using, which was NMR, by an unsupervised analysis, but we were also able to see that in the supervised analysis, we were able to differentiate them all. Should be noted, again, this was a pilot study, small sample sizes, and these were 3 replicates from each one of the cell lines. So looking at this data, we were able to differentiate pathways that could define differences not only between tumor normal, but also normal and unaffected, and normal tumor and unaffected. And so we can see that the pathways that were involved in the differentiation of these groups had to do with aminoacetyl tRNA biosynthesis and signal transduction of amino-dependent mTOR-C activation. So keeping this in mind, we also evaluated the different variants of beta-catenin. So as I mentioned, we had T41A and an S45F. And there were distinct metabolite differences in these two cell lines. as listed, 1-methyl-nicotinamide and the p-methylhistidine. Both have both have biological implications listed below, with NAD metabolism and then the beta-alanine and histidine metabolism. So from the pilot data, we were able to conclude that there was suggestive evidence that not only there were metabolic differences between desmoid tumor and its own matched fibroblast, but metabolomics was sensitive enough to differentiate the differences between these two variants of beta-catenin. This was a small study, but it was intriguing. So following the publication of our pilot data, there was a study evaluating pancreatic ductal adenocarcinoma metabolomics, and they noted that PDAC was able to fuel the TCA cycle through alanine secretions rather than glucose or glutamine. So like desmoid tumors, PDAC is driven by mutations in the Wnt pathway, KRAS in particular, and it's known to actually accumulate beta-catenin. PDAC is known to be a stromal disease, much like desmoid tumors, and so we hypothesized that if it looked like a duck, if it sounded like a duck, maybe it could be a duck, and so maybe we could therefore see these alanine secretions in these desmoid tumor cell lines as well. So we wanted to further evaluate the energy metabolism differences between T41A, S45F, and matched normal fibroblasts. So we employed a technique called stable isotope resolved metabolomics. It's a mouthful, but really important. And we're able to use the stable isotope C13 in our cell media in order to track the metabolism through these primary cell lines. GCMS was the technique that we decided to use for this. It's incredibly sensitive, and our collaborator, Dr. Guofeng Zhang, also at Duke, designed a targeted panel specifically for quantifying these metabolites in TCA and glycolysis pathways. So it is also important that we continue to evaluate these differences. T41A is a more common variant for desmoid tumors, but we also know that S45F has been noted to be more aggressive and reoccur more frequently. So this is not the same kind of metabolomics that we evaluated previously. So while we use metabolomics in our pilot study, it was an unbiased analysis of metabolites that were visible by NMR spectroscopy. This study, we used the flux analysis, which is metabolomics as well. We're still looking at concentration, but also how the C13 molecule from the cell media is incorporated into the metabolic pathways. So we can think of metabolomics and flux as you would with traffic. You can see on the upper panel, the car volume differences are equivalent to concentration differences in the metabolites. And the flux would be how quickly the cars could travel in a certain area. So we're interested not only in the differences of concentration as we add in these different C13 labels, but also how much flux there is through the pathway that we're going to be able to identify with the isotopes. So I have highlighted the glucose and the glutamine on the TCA and glycolysis pathway. And then also, to remind you, we are keeping track of this alanine and NAD from our previous data and from our hypothesis. I want to say a big thank you to Vijitha Kuvanar and JJ Bao at Duke for carrying out all of this laboratory work. None of this work would have been possible without them. The primary cell lines were derived from specimens that were collected at Duke and at 6 Kids by Dr. Allman under an overarching IRB protocol. The beta-cat status was determined and confirmed by Sanger sequencing and Western blot. In total, we have 4 matched T41A and normal fibroblast cell lines, and then 4 additional S45F. In order to carry out these experiments, we needed to do an incorporation experiment in order to make sure that the C13 molecules were being incorporated into the pathways. And that was determined to be a 6-hour time point. To date, we have collected GCMS concentration and flux data on the T41A and matched normal fibroblast cell lines. You can see on this slide that we have supervised analysis for the C13 glucose concentrations and incorporation. These are very good supervised models. The unsupervised also look good. There is good differentiation between the groups. And we can see that the metabolite concentrations and isotopes are different for the normal versus the T41A. Similarly, we also have good supervised models and unsupervised not shown when we incorporate C13 glutamine into the cell media. And you can see that the metabolite concentrations and isotopic differences that were significant for building these models are different, but equally intriguing. So unfortunately, because of COVID, as I'm sure you all have experienced, our progress on this research project was stalled. We are currently waiting for data for the S45F glucose and glutamine experiments for GCMS. The Duke lab just opened a few weeks ago. We are going to tackle all of the pathway evaluation and isotopic flux when everything is in hand. Some of the things that we are going to be considering as we do this analysis, both for the T41A and the S45F glucose and glutamine data sets. So glycolysis is increased twofold when we have proliferating fibroblasts. We know this, we saw this in our preliminary study. We also know that proliferating fibroblasts use glutamine for anaplerotic flux, while quiescent fibroblasts, on the other hand, will have these intermediates from pyruvate and oxalate. So we're going to be looking not only at the concentrations, differences in the TCA cycle, but which way the isotopic distribution seems to be favoring. So none of this, again, could be done without desmoid tumor patients and caregivers who generously donate to DTRF and provide materials for this research project. Again, I'm indebted to the staff at Duke and SickKids for all of their hard work. I appreciate your attention and look forward to our discussion at the meeting. Thank you.